I'm going to talk about integrating technologies and their application of these technologies in cultivar development, because in my role as a plant breeder, there are lots of avenues, there are lots of opportunities and lots of avenues to integrate technology. I am going to present a little bit on the AI Institute that is led by my colleague, Dr. Bhaskar Ganapathy Subramanian, and then also another colleague of mine, Dr. Shamik Sarkar, who leads the Translational AI Center at the Iowa State University. I work very closely with both of them. So I'm going to talk primarily about my projects later on, but I want to start by explaining just briefly about AIIRA, if you don't know what that is. In the US, there has been investment from the federal government, National Science Foundation, to set up AI institutes. And AIIRA is one of few AI institutes that have been set up in agriculture. And the primary idea there, one of the things that AIIRA works towards, is to create a digital twin that can replicate, mimic the physical biological system along with its virtual counterpart. And that is coming where we are assimilating a lot of data that comes in and integrating domain knowledge that comes from physiology, agronomy, biology. So making sure that we are able to come up with this digital twin that allows us to study agricultural phenomena, and particularly in terms of resilient agriculture. And we can ask some of the questions, testable hypotheses, potential solutions, avenues to mitigate different risks that exist. This is a representation, of that project where, as I said, it's integrating data that's coming in real time along with domain knowledge. These are model dynamics. And so the things that we are looking at, they include individual plant scale, but then also to the field scale. We're looking at plant breeding applications, but we're also looking primarily on the crop production that impacts farmers through the work that we are doing. As is the mandate for any AI institute, there are foundational questions that are being addressed in this project, but some of the outcomes of those foundational AI advances, they relate to the application that I, as a plant breeder, and the team that I'm in, we can utilize. I have to mention that there is a big human science component in AIIRA, because we want to make sure that not only are we building and deploying these digital twins or different AI tools, but that also there is an adoption that is happening to make sure that what we develop actually makes it in the hands of people that want to utilize it and make something meaningful from it. Of course, as you would expect, there is a big focus on getting the workforce development that would be expected in any large grant that is funded. Those of you who have interest in learning more, this is the website. It's just aira.iastate.edu. As I said, that for this AIIRA Institute that we have, the core technical thrust are building the digital twin, or let's say, just think of AI tools, then deploying them, adopting them, and then we also have a lot of interest in democratizing them. So we want to be using open source environment because we want it to be, the community can come together, so it has its, life cycle, that it continues growing and developing. And then we have these overarching themes that are in education and workforce development. There are, programs that we have developed in cyber agriculture systems, in minors, and, so some of the degree programs. And broadening participation, some of the things that my colleagues want to talk about, and then collaboration, knowledge transfer. I want to make sure that I talk about this particular project as well, because this is led by Dr. Shamik Sarkar. This is an NSF project that is on cyber physical system. It's a frontier project. These are very rare. Projects that are handed out. It's not precise. And this is being represented here. If we utilize the principles of cyber physical system, including individualized sensing, so all the way from individual plant scale to individualized modeling and reasoning, and then having actuation, which is one of the things that my colleague, Dr. Ardi, is going to present in her talk. But if we utilize the cyber physical system concepts, then we can move from a conventional agriculture to a more sustainable cyber agriculture systems. And in this, you can see that the difference is that here, we have integration of technology where we are moving away from these large, scale, inefficient, more capital intensive, to a smaller scale that is lower entry, barrier to entry, It can be more affordable because we are working with smaller units. It's more precise. We are going to individual level. I'm going to give an example of that later on. It is scale agnostic. If someone wants to run it on a large farm, it can. But if you want a small farm, you're a starting farmer, it can work on it. It has minimal safety issues because these are smaller units. So if you can distribute your task. But the most important thing in my mind is that there is reduction of cost, chemical cost. There is an environmental component because you are very precise in how you're going to manage these different pests, these diseases, because you're utilizing these technologies and ML learning that is coming in. And that's one of the focus areas in this project. I'm going to move a little bit more about some of the work that is happening in my team. So just to set the stage, this is what we do. I'm going to talk about very briefly. This is how we do it. I'm going to give some examples of projects that are in our group. Some of them are published, some of them are upcoming. And then how do we, or who we work with, who we are, and how do we make it happen? So that's sort of the outline that I'll follow for the rest of the presentation. In terms of the what, we are a program that is mandated by investment from producer groups to develop field-ready cultivars. That means that the cultivars we develop, it's a public institution, but the cultivars we develop, there's a market demand, the industry has a market demand for it, and farmers are looking to get the cultivars from us. So they have to be field-ready, and if you have background in plant breeding, this would be common for almost all breeding programs. You have to have good yield, economic yield, you have to have ability of the plant to tolerate different stresses, it has to have lower input cost for it to be profitable. Our program, this is an example of our soybean breeding trial. So you just have a visual of how it looks like in our fields. This here is a representation of a millet program that we have started more recently. And as you can imagine, the scale that we work is quite large. So we have extensive data collection campaigns. These are time series data, lots of different sensors because we have interest in a lot of different traits and they're in a lot of different locations. That sets the stage for the projects that I'm going to talk about, because why do we need to come up with, utilize these technologies for the work that we do? We have a diversity of projects. These are just some examples, some representation of different scales. We have ground robot, drone-based phenotyping, satellite phenotyping. I'm going to give one particular example. So we use in different contexts, but for time, I'm going to go through just one example each, and then multimodal, multi-platform phenotyping. I'm going to give some examples because our group has a lot of interest in water stress, heat stress, and then root traits. And then some of the work that is happening in the space with digital twin, particular interest we have in the augmented and virtual reality. And then my colleague's going to talk about the work she and Dr. Bhaskar are leading. So for our first example, this is an example of a project, ground-based yield estimation. Next set of slides are going to be arranged that this quadrant will have project motivation. This quadrant will have methods and analyses. This quadrant will have results. And then this quadrant is going to conclude for that project and the impact. So our project motivation here is that because of the scale that I talked about, and it's not just our program, any breeding program, the scale is quite large. So we have to phenotype at a lot of different locations, a lot of different plots. So we want to get away from taking the combine to a lot of different places because that is a 2-month-long process. We have to take yield from all different plots, and only after we run the combine, we get data. We have done projects on prediction, but we want to get as close to estimation as possible. So one of the motivation is that, can we do that? So this is just, very briefly, because I'm going to go through several projects. We are imaging on a running robot. This is an example of it. There is data annotation that we do in order to train the ML algorithm. It is doing individual plant tracking, through the plot, and it's also doing part detection, and then it is doing the estimation. That's the ML pipeline that exists. This video is an example of the algorithm is looking at the plant that it sees, and it's not taking into account the other plants. So it has to be able to see. So it's running in between the row, and it's doing the putting the bounding box automatically on a running robot. to tell us that, okay, these are the number of pods that I have, and in soybean, we can do some estimation of what would that correlate to yield. In terms of ranking, our results came up whether we took multiple frames or a single frame. The difference was not that much. And what we showed in this paper that's published is that for a breeding perspective, it works because we can do effective ranking to make correct decisions to advance the ones that have merit to be advanced. It saves a lot of time, from a breeding context, and also resource, and it also provides efficiency. So this is one example of a ground-based robot. For drone-based example, I'm presenting the work on maturity. So because of the photoperiod sensitivity in soybean, there is a lot of, a large range in soybean maturity. It is very difficult. Generally, about 10 people are going to spend about 1 month to take maturity ratings every second day. So you can imagine the amount of time it would take, to get that idea as to what is the maturity, because we want to compare similar maturities to its yield, because otherwise there are biases. Plus, we want to know the maturity because we have to decide which region of the US, this particular soybean variety is going to be effective in. So this is the motivation here. This is an example, so you can get, some of the idea of the scale. These are plots that you can imagine, if your job is to go and, go there every day or every second day, because, different plants are going to mature at different time points, it's quite extensive. So we've used over 3 years of data, more than 10,000 plots, 8 time points in order to develop our model. And we tested to see whether we could do, so the rating is generally done as a 1.8, 1.9, 2.0, 2.1. So we looked at every single relative maturity. But what we found is that our best result, which is very good for the program, it comes out that if we put categories of maturity group 1, maturity group 2 early, maturity group 2 late, maturity group 3, and maturity group 3 late, we get really good results in F-score because we wanted to use both precision and recall because, we want to make sure that we are, getting the full metrics. It works out quite well. Now, what does that mean? That means that if we take drone data, and this is accumulation across the time series, for that particular plot. So if we take drone-based maturity data, we can do that work in about 8 hours of flying time compared to what I said, about a month of 10 people, going over time. Great use of technology that, simplifies things, for us. If you have interest in some of the work that we have published in terms of more review articles, this is a paper that came out because a farmer approached us, and that farmer said that I've yet to come across, I fly drones, but there's no paper I can find that explains, what is happening and what should I do if I am interested in drone-based phenotyping. So based on that motivation from that farmer, we published this paper a couple of years ago. And then more recently, this is very recent, this is cotton, maize, soybean, and wheat breeding. It's a review article that's come out in Crop Science just like a couple of days ago. The third example I'm giving is a satellite-based phenotyping. So the ground I explained, drone I explained. Now we're moving to the satellite scale. You can imagine with drone-based, it still takes time. In satellite imagery, there are advances. That resolution has become quite good. This particular satellite that we're using, it's a 30 centimeter by 30 centimeter per pixel resolution. So for our plot, we were able to utilize, so we're interested in crop health monitoring, we're interested in yield estimation or yield prediction. So we are using multispectral data. This was a 5-band from the satellite, the LIDAR data for the wetness index on the soils, on the topology. And using different features, we were able to see that it's not perfect. There are only about 47% of the variance is explained, but correlation is 0.62. That's just the first attempt of utilizing satellite-based phenotyping on more than 10, 15,000 plots across different stages of the program. So it shows a lot of promise for us. So instead of coming up and aligning and making sure that the drone is flying over, because our field sizes could range anywhere from 1 acre to 10 acres just for one experiment that we have. This is an example of one time point. We're getting the data that we need in order to do that yield estimation or yield prediction. You can see that impact, is quite significant, in my opinion. So we are excited about it. The other thing that we are interested in is in plant breeding trials, we try to account for heterogeneity that exists in the soil. So all of the work that we do is in the field. And you can imagine if just in this room, if this is your field, same variety is going to look different in this field. And that's because of some of the differences in the soil characteristics, among other factors. Plant breeders for decades have been using adjustments to account for that heterogeneity that exists. But what we did not know are what are the soil features, and can we use the soil features in terms of understanding and then accounting for that adjustment that is happening. So that is the motivation here, that can we develop an ML-based spatial adjustment and quantify soil properties effects on yield. If this works, that means that not only can we have better spatial adjustments, it means that we will be able to explain those adjustments. And then we can also see that different parts of the field, what specific nutrient stress exists, because that is an opportunistic selection that can be done. So this particular figure that you see, this is an unadjusted example of a particular test. And after spatial adjustment using, this was a extreme gradient boost method, you can see that we are utilizing different soil features, and we are able to come up with better adjustment. This is now gone. But how do we make comparisons that this method actually is working? So we used Moran's I, and closer it is to 0, better it is. It probably is not visible, but this is the XGB local. That means that these are breaking up the field into smaller. This is XGB global, which didn't do as well. And this is what the current state of the art is. It is quite far away from being optimum. So this is the closest. Looking at similarity coefficient, that is saying that they make similar kinds of decisions. And then again, we see that the XGB local does the best job. In terms of mean relative efficiency, because these are unreplicated plots, we have to use checks that are replicated to see that which of these methods have the best relative efficiency. Again, we see that this particular method has the best efficiency. So what I'm saying is that integrating soil features, it makes for better ability to account for heterogeneity. We can make better decisions of what to keep and what to discard. And then we can also use this as Shapley values. We can then explain what was happening at that, in that plot, because this is combining digital, I should have mentioned, this is combining digital soil mapping. These were soil cores that were taken, and then ML algorithm is used to create that digital soil map. And we are integrating ML model to utilize those features along with the data that we have. So Shapley values explain which of those features are the predominant driver of that variation. So now we have explainability along with adjustment. Plus, because we know that it's particular nutrient stress that is inherent in that part of the field, which we would not have known without these digital soil maps and this algorithm, now we can make more opportunistic selection for nutrient stress. This here is an example of utilization of different systems. This is a drone-based system. This is a drought nursery. So you can see that some plants are dying and some are not, which is very good for us. That means there is genetic variation that exists. Drought is something that we are mindful of. People may think that Midwest does not have this problem. I think this is true everywhere. We are getting encountering more and more. These are just maps of different years. So we are using hyperspectral data, thermal data, multispectral data, RGB data. And when we look at these, create these different indices, it is very clear that we can parse out the wilt score, which is the rating scale that we use to determine which accessions are tolerant versus susceptible. So it's doing all good. One of the challenges that no one wants to, or I don't want to be us to collect that, many data, so many different sensors. So what we are using is a self-supervised learning framework, where the first step is to use multi-MAE, which takes data from all of these different sensors, combines it, and learn the cross-model representation. And then we utilize the encoder block to get the wilt score prediction. What that means is that instead of utilizing all of these sensors, one of the sensors could do as good a job as we would get from these multiple sensors. So that's the direction that we are going in that project. This is an example of a heat stress tolerance project. Again, heat is one of those things that People may think warm season crops are not, they're fine, they should be fine. They're not. There is a threshold. Beyond that threshold, even warm season crops do not like, because they do not have the physiological response to handle it. So we are interested to figure out what type of variation do we have for heat stress tolerance, and what can we do to make better selections. So this is an example of a study that was done indoors initially to come up with a heat stress response index of the treatment in the heat stress chamber versus the optimal chamber. And what we found out was, that makes sense, that canopy temperature in heat stress, it's higher. And these are, this is a study of almost 450 different unique accessions. And then in the normal temperature, it's not. Metal conductance, it's lower in heat, it's better here. Quantum efficiency in light, lower here. All of this is expected. We expected that dry shoot biomass would be lower in heat compared to optimum, but we didn't find that. In fact, we found that it's a little higher, which is interesting. But in terms of genotypic variance, we found that for heat, we have genotypic variation for all these traits, and for optimal, and then also for the index. That's good. From a breeding perspective, that means that breeders can utilize the germplasm that we have screened in order to make improvements for heat stress. I want to quickly show, this is a rapid speed. Heat stress, the challenge is that it is very hard to do heat stress screening in the field. So what we wanted to create were these mobile greenhouses that can be constructed and placed in the field, and they have the ability to maintain temperature the way we program it to. So in this video, as you saw, these students have created this, I think it takes about an hour to hour and a half to set this up. You can leave it outside for however long your treatment, calls it for, and then you can dismantle it and take it away. So now this is, one scale, but that means that if we wanted to increase the scale, we can, and now we can do some of these heat stress studies in the field. This is a great project example. This is mechanical engineering students who do a capstone project. So more than 100 mechanical engineers, students in their final year, have worked with our program for different projects. This particular team is working on heat stress chamber. They're given a project objective. They work with us to develop solutions. So it's a great example for them to understand agriculture, great way for us to integrate them to think about solutions for agriculture as well. And this is just an example of that chamber outside in the field that we have demonstrated. I'm going to shift to roots-based phenotyping. This is something that is quite dear to me. It's very hard. Is anyone in the room that works on roots? Oh. That's fantastic. Okay. I'm so excited to hear that. So, as you know, root is a very difficult trait to work with, right? I'm fascinated by it. I love it. But I think there are opportunities to, look at some solutions from a breeding scale. For a physiologist and agronomist, where you're working with smaller population sizes, smaller number of samples, it's doable because you can do that extensive measurement. From a breeding perspective, where our scale is several hundred, 100,000, or at least several, in tens of thousands, it's very difficult for us to, get through a lot of different accessions. So our motivation was that can we come up, and this was done in controlled environment conditions, and we come up with utilization of computer vision and ML with a program that gives us these characteristics of root, and we wanted to see what type of diversity do we have in the soybean germplasm. So that's the software that was developed, and these are two papers. The first paper is a methods paper, explains what AIIRA is and how it was developed and what kind of traits it can come up with. So there are more than 35. Some are more relevant than others. And then there was a second follow-up paper that utilized different types of analyses to essentially study the diversity that exists in characteristics. This is a great visual, at least from me, because it shows that if we combine, because from a breeding perspective, we wanted to come with an index, we wanted to come with an idiotype. So these are informative root types. We call them I root types. These are just, you can see features. This is deep, steep. These are an umbrella type of shape. This is nutrient foraging, so they can set up their roots so that they have maximal opportunity to take these soil immobile as well as mobile elements. And then there is a maximum type that we developed that can we maximize it? It's not a physiology experiment. I have to clarify. This is just for our sake as a breeding perspective, that what type of variants exist. And then it has to be that which one of this is, what is needed in the context that we are working in. The other one that we were quite interested in was nodule phenotyping. This is again a very difficult trait. So, if someone asked me that how many nodules are in this particular route, that would be a very difficult task. If that was my job for a month, that would be a difficult job. You can imagine that these are the nodules that were taken from this route. These are the nodules, they're arranged with different root sizes. Those are a lot of nodules to be collecting, right? So we wanted to come up with an automated way of getting total count, and then we're interested to see what else could we pick up. So essentially what this model is that it is able to come up with the total number of nodules, and it is also tracking taproot, so we can determine nodules on the taproot and then nodules on the non-taproot. What this so this paper came out a little while ago. It's called SNAP if you have an interest in it. And then there was a follow-up paper that essentially is exploration of the type of observations we got from more detailed investigation of fewer genotypes. So we can get nodule count, we can get volume per nodule, we can get total nodule volume. So nodulation is not just total nodule count. We can have nodulation that integrates the nodule count as well as the area or the volume. It only works for these oval-shaped nodules for now, but this is an example where some of the questions we had, because we wanted to come up with the best root type, as well as best utilization of nodulation, because we don't want to maybe stress the plant that it's producing too many nodules, nor do we want to have too little, so it does not meet the requirement of the plant. There were some interesting observations in terms of taproot nodulation. There was a good correlation with percent nitrogen in the seed, so something that needs to be investigated, that it's some of the critical stages. So this particular project is augmented in virtual reality. None of us can be everywhere at all times. That means that there are missed opportunities because we cannot be able to phenotype wherever we need to be. So what this project is looking at was using terrestrial laser scanner to construct the 3D point cloud. We are now moved to a 360 camera for stereoscopic videos. And this was in a headset environment. So this is what it looks like. This is immersive, where we can use it as a training. This is a field of iron deficiency chlorosis screening. You can see that there are some healthy types and unhealthy. We can use it as a training module where a new person can go in and they can click. They have the headset on, they can click, they can understand which particular rating is reflective of what experts have rated. But it doesn't need to be just for a training. This is something that if I cannot be or someone else cannot be in that nursery at the right time, if we have this information, then we can utilize to make selections. This here is an egg gym, so gym environment where it's a deep reinforcement learning. And one of the things that we learned is that crop modeling is doing a really good job in terms of growth and development, but there were very few instances of opportunities to learn about the spread dynamics of pathogen and to mitigate in a timely manner, in an effective manner. So this particular example is to try and come up with ways to see that we can simulate conditions that is using the reinforcement learning to determine where the pathogen starts, and based on the weather variables, based on other parameters, the topology, the variety that you have, to come up with what kind of pesticide should be applied, at what efficacy, at what level, at what time, and very site-specific spray, because that fits in with some of the work that Arti is going to talk about. So this is, again, very exciting. It seems like we used some real-life example to show that this actually worked, that real-life satellite-based imagery of the spread dynamic, this RL was able to come up with it, although it had never seen that type of data. So I'm going to try and bring it together now in terms of who we are. So we're very fortunate that we have opportunity to interact with farmers, to learn what are the problems that they're encountering. So whatever problems they're encountering, there is a lot of parallel for the plant breeding group, so we can utilize the same types of solutions that exist for crop production. into our plant breeding program. And we not only are we interacting with them to learn problems, we actually interact with them to talk about potential solutions that they would be interested in that they could apply. We have had farmer leaders, be panel members for our machine learning for cyber agriculture system to come up with, that problem formulation and work collaboratively with them. I'm very fortunate to work with, great group of researchers, part of the Soynomics team. And then this is an image, or this is a photo of a team meeting from last year where, we come together, the group, and we talk about different projects that are ongoing. And our collaboration, they exceed Iowa State. So we have outside Iowa State collaborations as well. So I've gone through the what we do. I've given some examples. This is not a non-exhaustive list of the work that is happening in the team that I'm in. I've talked about how these projects are coming together because of a great expertise, my colleagues and other students that have. One of the things that we learned was that we're very excited about the work we do, but it is very difficult for getting the information, or the public may yet not know what we are doing. We talk science, but it's, we want to make sure that we are engaging, general public. So what we have done is we have created these videos, sets of videos, some projects that we picked, and they are on YouTube. So wherever we get a chance, we can share with them. These are students. The PIs are not in the video. These are students talking about what they're doing and why is it relevant to, the general audience. And they've had very good reception from the general public. In terms of moving forward, I think crop improvement, plant breeding, cultivar development, some of the things that we are talking about, decades ago, problems existed. Decades from now, problems will exist. So there have to be, bidirectional advances that happen in crop breeding and crop production. I think they do coexist together, because these problems are quite variable and they are complex. Climatic resiliency, one of the things that AIIRA is focusing on. It requires, innovative solution, out of the box thinking. And that means that, these are some examples that I talked about, but that means that we are going to have collaborations across geographies, across disciplines, truly working together, because these problems are way too big for just one or two disciplines. And with this, I'll conclude by acknowledging these are listed alphabetically in each of the order for federal agencies, farmer-led groups, institutional support to our program, again, the PI team, and then again, the research team. And there is this book, Plant Breeding and Cultivar Development, that's an output from when everything was locked down in COVID. My colleague and I, we finished this textbook. So if you have an interest in plant breeding, you can take a look. With that, I'll conclude. Thank you.